Good morning, everyone. This is a tutorial on accessing sequence read archive data. It's really more in a tutorial spirit, technical aspects, not really a talk. It's in a spirit of a different talk I did in 2022, and this is sort of a screenshot of that video during that talk, where I was commenting on CGSI format, where some speakers give two talks, and at the time I was saying earlier, even 2 years ago, so in 2020, I was giving two talks, and my second talk was more not absurd, but more original than the first one, and it became more successful in terms of view counts in 2022. So I was commenting on the usefulness of doing more original second talks. And as it turns out, this talk from 2022 also became even more successful than the other ones. So there's something to be said about making second talks at CGSI, which is probably better than the first talk. So if you didn't like the one yesterday, you might like this one better. Let's see. So in that spirit, I'm actually doing the same format as 2 years ago. So I'm doing a tutorial and live demonstration of sequence bioinformatics software. And last time it was pangenomics with graphs. This time there will be no pangenomics, no graphs, but hopefully still interesting to you. It will be about accessing this database that I've been talking about for many years now at CGSI and elsewhere. Sequence Read Archive at NCBI contains all of sequencing data from about 25 years ago. It's now 50 petabases in size. This is the helpful table for knowing how much a petabase is. That's a lot. And it's been growing almost exponentially for the previous years. You can access it through NCBI or ENA. And it looks a little bit like this. You can search by metadata, but you cannot search by sequence. You can download reads, but it's a little bit convoluted to download reads. This is really the point of this tutorial, to walk you through accessing this huge database. And as a motivation, this is a map of all samples that have been uploaded to SRA across time. And this is a heat map. You can see that pretty much the message here is that it's samples all across the globe, even the oceans pretty comprehensively, and of course some continents are more sampled than others. So for this reason, my colleague has very grand words about this database, and Artem says that it's open science at its best, and he has this 3 lines quote that I could not come up with it, but I guess he did. And he says that this database is a reflection of Earth biodiversity. It's a shared knowledge, of shared heritage of all living organisms. And we are, as bioinformaticians, responsible for liberating and sharing this heritage with everyone, freely and openly. So I cannot understate how important SRA is for biomedical research and biology research. And even more striking is the fact that this data is free and accessible for free for the rest of time. And this is because in the US, there was a consortium called INSD that has stated that all the data that will be uploaded to INSD databases, such as SRA, will be free and unrestricted in the public domain. And because of this consortium, we have this resource. Otherwise, there is a timeline where SRA is protected, and you need to register, or even you need to ask for access. And thankfully, we do not live in this timeline. We live in a good timeline, the one where SRA is open. And for this reason, Artem and others I guess mostly Artem, calls it the Library of Alexandria of genetics. So hopefully this is convincing enough that accessing this database is a privilege, and how do we do it now is the purpose of this tutorial. So today, it's a tutorial on accessing SRA, and I'll show you in real time some ways to access it. Perhaps to go a little bit technical as to why and what's in there, it's mostly raw data, raw sequencing data, short reads, and some long reads as well. And accessing this is useful for reproducibility, reanalysis of the data, probably many other reasons, searching for particular sequence of interest. And first of all, access tools provided by NCBI mainly are great, but some techniques are not so well known. And also, I've developed a project with other people since the last few years called Logan. I briefly mentioned it also during this talk, and it greatly simplifies access to the SRA, and I'll show you how. SRA has been mirrored to the cloud about 4 years ago now, and this is revolutionary because now you can access all of the SRA reads, and especially all of its metadata, and this is a bit less well known, through means which are faster and more convenient. Before, the data was sort of quote-unquote only mirrored at 3 locations in the US, in CBI, in the UK, ABI, and in Japan, DDBJ. Most of the commands I will type today, so it will be recorded on YouTube, but also most of the commands are at this URL on GitHub. It's a slightly different tutorial, but it contains the same spirit. And the slides for this particular talk I made publicly available at this QR code and URL if you want to follow along. Part 0, let's start with the metadata of the SRA. If you want to search for some data there are multiple sort of levels of difficulty. I guess level 0 is you look at a paper that has already been published, you scroll down to the data availability section, and you pray that there is an SRA accession. Not always the case, but in some papers, such as this one, there's not only one, but there's a good amount of them. And all of their sequencing data has been properly uploaded to SRA, and they give you the projects. So you can basically visually grab for SRR something, and you get accessions for the run level. There's other ways, so perhaps you do not look for a paper in particular, but you look for a certain collection of data sets that follow some keywords, then in that case, you can search SRA website with the metadata in a search bar. Sometimes it works, sometimes it doesn't. For instance, in this example, I'm searching for chicken, and the first hit is Campylobacter coli. I'm not really a biologist, but I can certify that this is not a chicken, definitely. So, well, it's perhaps a funny example, but in other cases it might work. But this is showing the limitations of searching by metadata. For some reason, the wrong keywords might appear, and of course, incompleteness of the metadata is notorious everywhere, but especially in the SRA. So there are other ways, and let's consider this one. There exists a cloud-based, as I said earlier, metadata mirror that supports SQL and SQL queries. It's a programming language for doing database queries that you probably have seen before. So here, if you want to access a large amount of metadata very quickly, you can formulate a small program in SQL language. And this one, I guess you can perhaps read it and then guess what it does. It selects all accession from the SRA metadata table that not only are public, but contain reads that are on average are longer than 31 base pairs. And those reads come from libraries which are transcriptomics or metatranscriptomics. Furthermore, apparently this is also OR RNA-seq. So this is a comprehensive way to select. You can also do it on a website, I suppose, but this is more versatile. You can select all RNA-seq data from the SRA in a few command lines, and I will just illustrate shortly how to do this. There's multiple ways. The database is mirrored at 2 locations, one on Amazon, one on Google. This is Amazon way of things. It's a service called Athena, and it's loading here. It's preloading the same query as I showed on the slide. You can click Run, and then it shows you how much time I spent for doing the query and how much data has been scanned in terms of metadata. And after a few seconds, you get a list of 7.8 million accessions from the SRA. And okay, this is a long list. You can actually download it here, download results CSV, and you get a CSV file. You can, of course, modify the query a little bit if you want to access other information, for instance, size of the data set, its basis, for instance, and run the query again, wait a few more seconds, and then you should get the same results, except that not only you have the accession, but also the size of the library as another column. And there are many other columns. They are described here in a metadata table, but it's not very explicit. I prefer to actually search on Google the description of each of these fields. If you type SRA cloud on Google, and then actually I've pre-cached the URL, but you can access it very quickly. It tells you what all of these fields actually mean. So accession is accession, Assay type is RNA-seq of others, and the one I was using earlier was M-bases. I guess it's here, number of megabases in the run. So it's a little bit small to look at it. I should actually zoom in. And there are many others which are interesting, such as organism. Okay, so that's one way that I like a lot. There's another way that I want to highlight is that the metadata that I just showed you has another table called taxonomic analysis, where you can search by taxonomic rank. And this is very powerful because this metadata is not human generated, it's machine generated from the data itself. So it cannot contain errors. It's the same type of errors, at least, that, I mean, I see David laughing. Yes, it probably does contain errors, right? But it's not the same type of errors as the type of errors humans make. Let's put it this way. I actually talked about this in my previous video in the Logan talk, but now I'm demonstrating how to access it. So level 3, searching SRA with taxonomic metadata. Here, in this query, I'm selecting accessions and a field called total count, I'll explain it later, from this particular table that retrieves all the accessions that have been annotated to contain a taxonomic ID of 4751, which corresponds to fungi. And total count above 100,000, it's roughly, so the technical explanation is a bit involved, but it's roughly a number of reads that are from this taxonomic idea. So I'm selecting accessions that contain significant amount of mushrooms. And let's run it. And I guess what's powerful about this is it doesn't rely on human-generated metadata. So you don't go to the SRA website and type mushroom. You actually pinpoint from the reads that have been annotated by a previous analysis accessions. And then you get 400,000 accessions. Then up to you to download those accessions and do further processing. And I will illustrate later how you could do this. But getting this list is already a good first step, right? Hopefully. Yep. You might ask how to set this total count value. Like, is there a way to set it? I asked Rayan Connor at SRA, at NCBI, is there a way? And he answered me this on Slack. There's essentially no way. It's more of an art. If you can't read it, it says that there is not really a magic number for the cutoff. It's currently a bit of an art. He explains a bit further that you could perhaps estimate the number of reads in the accession, and then sort of do a ratio. Like if you don't have lots of reads, then total count should be small, and if you have lots of reads, you should have more. But there's no automatic way. So I like 10,000 or 100,000 as a first pre-filter. Then you can combine everything. So this query now gets all SRA metadata for accessions containing high abundance of a particular taxonomic group. So you combine two tables. One is the metadata table that contains information entered by humans, and the other one is a taxonomic table that contains this tax ID and total count information. So I think this was generated by ChatGPT, but it just works. This is a SQL query that gets all the metadata that I like for a particular taxonomic ID, for total count above a certain threshold, and where on this metadata table things are public and long enough. Let's run it. One thing to notice when you run such a query is that it's going to scan way more data because it's actually scanning 2 more tables. It's a bit longer query. Already about 50 gigabytes of data have been scanned. It's very efficient. That's the thing about these cloud-based queries. And now you get about 1,000. This is another tax ID, I think, for a particular bacteria. And you get a rich set of metadata on it, not just total count, but everything that has been entered by the submitter. Okay, and you can, of course, download all of this. I will not do it now. Oh, okay. I show you stuff on Athena. There's also Google BigQuery that supports the same sort of operations. In terms of cost, I've noticed that Athena is about $5 per terabyte scanned. So the number of bytes scanned you saw earlier actually matters. And typical scans of SRA metadata range from 200 megabytes for simple queries to about 100 gigabytes for more complex queries. Google has an interesting scheme where they can actually do 1 terabyte of query for free per month. So interesting if you want to go in free type tier territory. So yeah, this was a demo, so I've run it at the same time. Okay, now moving on to reads download. Oh, question. Yeah, so the question is, do the metadata get updated at the tax ID level or accession level? I think mostly for both not. For the accession, surely not. Like once it's submitted, it's the same metadata stays. For the tax IDs, it's up to the stat group at NCBI, and as far as I can tell, they have not updated. Because processing all of this is extremely expensive. So they did it once, and they do it for the for the updates. If a new accession is uploaded, then they process it using the latest version of their pipeline. I don't think they process the previous versions again. Okay, I'm going to go a bit quickly on this because there's more material. Downloading SRA reads. So once you have the metadata, it's a good idea to get the reads corresponding to these accessions, right? How do we download accessions? When probably most of you have tried downloading from the web. You got to go to the SRA website, you click somewhere, download FastA or FastQ. And I've done this for one particular accession. It takes about 2 minutes. It's a small accession, a few hundred megabytes. It's okay, it's not too long, but if you want to download many of them, like 400,000 results, it's not practical because you will need to click one by one. Also, it's quite slow. You've probably also tried the command fastqdump provided by SRA Toolkit, and if I do it on the same accession, turns out also 2 minutes. I like this technique that is actually recommended by NCBI, where you actually pre-fetch the accession first, and magically, if you do that, it's a bit faster, because the toolkit from SRA is a bit fragmented. So there are many tools that do overlapping functionality. FastQdump is sort of the legacy version. Now you can do pre-fetch and then faster QDump to do the same effect, but faster. So pre-fetch takes about 28 seconds on my machine when I tried it for download, and 4 seconds to extract. So what this does is download everything, mainly the SRA file, which is a compressed representation of the reads, and then converts it to FastQ. This is faster QDump. And finally, the one I want to highlight is a bit more convoluted, but it's actually faster. There is a way to access SRA data using its cloud mirror and perhaps directly guess where the SRA file is. So it kind of bypasses what prefetch does. If you type this command line, it will download the SRA file. When you move it to add the SRA accession, the SRA suffix, and then you continue with the same as before, faster QDump. If you do this, it takes about 15 seconds to download, and mainly what the time saved is that prefetch does some queries to the NCBI servers. None of them happen here. It's just directly downloading a file from a S3 bucket. However, for 15% of accessions, this clever scheme is not so clever, because faster QDump will actually download stuff in the background without telling you. Because of the compression system used by SRA, some of the accessions, about 15% of them are compressed with a reference, and you need to download the reference to decompress the read. And this is done by faster QDump. Prefetch actually does it behind the scenes. So yeah, just something to know. Technical dependencies, so I haven't really talked about it, but S3 is like a Dropbox type storage hosted by Amazon. It can be accessed using a toolkit called AWS command line interface, but there's also ways to access it with just wget on Linux. And similarly, SRA toolkit is fastqdump, faster QDump, is a set of tools provided by NCBI to access the SRA. Later, I will mention 2 other tools. Z-Standard, ZSTD, is a compression format like gzip, and KAT is a genome assembly analysis tool from Earlham Institute, I think, in the UK. I'll probably skip the demo on this. I have 2 more parts. Any questions on the reads part?
It's annoying that FastQ doesn't output a GZIP format.
Oh, perhaps it doesn't, yeah. Right.
So the SRA file is small.
Right, SRA file is compressed, compacted at least. FastQDump probably doesn't output GZIP. Faster QDump, I'm actually not sure. I don't have it, huh?
You can download FastQGC with Raspberry.
Yeah, Raspberry,
So what I like to use with faster QDump is, well, you can do -- faster unsorted, so that you discard FastQ qualities. It's already some time and space saved. And then you output to std out, and then you pipe it to Z standard for compression. I did not. Right. So actually this could be level 5, but I didn't even mention it. But good question. Thank you. Yes. Oh. Not that I know of. You can really do a lot of AWS queries at the same, as much as your machine CPU supports, then you can do it across multiple machines. That's what we did in Logan. So, yeah. Okay, moving on, Logan contigs. So it turns out, instead of having to download SRA reads, you can just download the equivalent Logan contigs, and it's actually much faster. So Logan contigs, I should skip to that first. What are those? They are short read assemblies of almost all SRA accessions up until December 2023, with planning updates of 24 and 25, but they haven't happened yet. It can be seen as a mirror of SRA that is 100 times smaller to download and to access, with a tiny bit of information loss. So it contains a good amount of all solid k-mers from the reads, and well, contigs are exactly what you think. It's an assembly of the reads. You can have more information at this URL, and I will quickly demonstrate how to access. So if you type this command on a terminal, or you know what, I'm not going to copy paste. The format is as follows. You type this prefix, which is URL of contigs. Then you type the accession actually twice. Oops. I'm happy to do a clear, yeah. Thank you. Then it always ends with contigs.fa.zstd, and then dot to say that you copy in a current file, in a current folder, when we've just downloaded the contigs. And it's much faster than downloading the reads, and it contains almost the same information. If you want to look what it looks like, it's a FASTA file. Wow. Sorry, I'm going to clear again. I'm going to display the first 100 characters. It's a FASTA file of the assembly with the header, some graph links. I will not get into that. Abundance of this config is here, and then the sequence starting here. Okay. A slightly more efficient way is if you, instead of downloading to the folder and then decompressing, you can download to std out and then decompress on the fly by piping to Z standard cat, and then you get a FASTA file directly. Note the dash instead of the dot. About same runtime, slightly faster even for some reason, because you don't write to disk. Well, you do write to disk actually. I don't know why it's faster. Anyway, was it 3 seconds? Yeah, it was 3 seconds. Randomness. And I guess I will just flash the slide on Logan that says that we've compiled this big data set. It's essentially a mirror of SRA containing all assembled sequence on Earth. There's a preprint, there is a CGSI video from last year. We've estimated the direct sequencing cost equivalent of SRA, which is about in the order of $10 billion of sequencing data, not even counting the human cost of generating this data, just the sequencer's cost. It's a huge resource. It's much bigger than GenBank or BlastNT. I've plotted this tree for a subselection of organisms that shows the amount of data you have per species compared to the amount that's present on GenBank. And for some, or at least a good amount of organisms, there's almost no data on GenBank comparatively. So you can see it as a Earth-wide pangenome reference, species by species. I'll skip. I will not skip that. It's interesting to verify the loss of information in Logan context compared to the original reads. And to do that, there is a cool tool developed a few years back called Katz. I'll just copy-paste those command lines. What Katz does, it compares the raw reads that I've previously downloaded to this folder with the Logan context. And first of all, it's doing k-mer counting on both. It's going to take about, I guess, a minute. And then it does a plot, a histogram plot. I was mentioning this in my talk yesterday, but I'll go in slightly more details today. For those of you who've done genome assembly, they may be familiar with this. If you're not, it's going to be, it's creating it. I'm going to show it in a second. Unfortunately, the tool is a bit old, so now it takes a bit more time to create. There's a newer tool called Mercury-Fast-K that does the same thing faster, but I couldn't install it on my computer, so I had to rely on the old tool. I guess I could install it, but then it used more memory. Fast-K. So it's a variation of Mercury developed by Gene Myers, who's created Fast-K, that uses modern k-mer counting with modern histogram analysis tools, Spectra CN. So yeah, now it's time to comment a little bit that plot. So this is a classic k-mer histogram plot that you may have seen before, showing the number of k-mers having a certain abundance. So this is a very clean sequencing data set that has about 50x abundance. Two things are interesting to notice. This red component corresponds to good genomic k-mers, and they are mostly all found in Logan context. That's great. And those black peaks correspond to sequencing error mostly, and you can see they are notated as 0x, meaning that they are not found in Logan context. So the job of the assembler has been properly done. So they removed, all the errors have been mostly removed, and all the correct k-mers have been mostly kept. And that's what you want. Any questions on this? Nope. I realize 30 minutes is actually very short compared to previous versions of pangenomic toast was 45 minutes. Quick note on bacterial data. Don't use Logan for bacterial isolates. There's actually another project called All the Bacteria that has higher quality assemblies. And for human genomes, there are some human cell lines in the SRA, but I would recommend to check out the Human Pangenome Reference Consortium for higher quality human genomes. And if you want to access control access data, such as disease cohorts and so on, you're out of luck. They are not public, so you need to request access, unfortunately. I guess this is what it is. Do we have time? Not a lot of time. I have time. Thanks, Zara. Cool. Part 3, Logan search. 1 second. OK. I thought it was worthwhile to mention it. At the same time, I will totally undersell it. I think it deserves its own talk. It's a very, very recent development. We now have a way to directly search the entirety of the SRA by sequence. So the way it works is that you query a particular sequence. Behind the scenes, there's a huge matrix represented here as a 2x4 matrix, but it's really a 1 petabyte matrix. It was a challenge to construct by Pierre and Theo. And as a result, you get a list of SRA accessions that contain your query sequence. So behind the scenes, it's as if it has searched 50 petabytes of data in a few minutes to tell you where your sequence is. So this skips metadata finding. This skips reads download. It even skips Logan context download. It skips everything. You just have a sequence. You know where it is in the SRA. You don't need to do any sort of cloud access of anything else. Let's illustrate it. You go to logansearch.org, and then you click Query the Planet. And then it will ask you to input your query sequence. Suppose you process an unknown sequence, you have no idea what this sequence is. Or if you do have some ideas, you would just like to find everywhere in the globe where this sequence appears. So let's suppose this sequence is unknown. When you click load, you put your email, and then you search a certain set of groups. You can search all of it. I recommend also sometimes searching all no viral, because if we don't care about viral sequencing, which is mostly coronavirus, it's a bit faster to query Logan search. Threshold, no time to explain what it is, but set it at 0.5 or a bit higher. Then submit. It will take about 2 minutes to run the search. It will email you when the search is finished. I have, in the next slide, the results from the same query that I ran yesterday. So I'll just show it now. And then after 2 minutes, it shows you this, which is a list of SRA accessions along with SRA metadata, the same one we saw earlier this morning, of accessions containing this sequence at a certain percent of found k-mers. So this first accession has 94% of all query k-mers and so on, in decreasing order of number of k-mers. And then using the metadata, what we did actually is an AI summary of all the metadata just to give sort of an overview of what this query sequence is. So here it seems that this query is likely from organism Procyon notor, also known as raccoon, and so on. You can plot a map of where this data in the SRA appears. So apparently those raccoons are found and sequenced in the East Coast US, and some in potentially Denmark and elsewhere in the world. We provide some plots automatically generated from the metadata as well. For instance, number of accessions for a particular organism. It's a bit hard to find, but there are some default plots provided at the bottom right. Let's take some time to plot. So it's mostly muscle tissue. Okay. Sequencing center, if you are ever interested in that. Yeah, Copenhagen and Smithsonian and so on. Finally, the last functionality is that you can actually have a real alignment of your query to particular accessions. Yes, question?
You have to do it only using web interface, or is it?
For now, it's only web interface. We're working on providing API. There are some limitations, but hey, this is the first interface in its category. So I guess for now it's better than nothing. But if people request, we're happy to go further and provide API for people who request it for now. Let's see if this works. Yeah, this works. So the query has been aligned to this particular set of contigs from this particular accession, and now you get a blast-like result that usually you don't get with k-mer queries. It's a tool, I think, provided by Pierre, called Back to Sequences. So behind the scene, it's actually running on a virtual machine, a download of the Logan contigs, and a k-mer search to find the actual contigs corresponding to the Logan search hits, and then runs, I think it may be a Smith-Waterman alignment, I'm not quite sure. Good question. But this one, I mean, as you can see, is very, very high identity. Okay. Don't want to go too much over time. So that was Logan Search, a very powerful tool, totally underselling it, but showing a little bit what it looks like. Feel free to use it. It's free for now. I guess. It's actually quite expensive to maintain and host. So in this tutorial, we've shown many ways to access SRA data through metadata search, read, download, Logan contigs, download, Logan search. Some conclusions: open data is great, metadata, it exists, it's sometimes imperfect. And Logan mirrors the SRA and makes it more accessible. Metadata, SQL queries to access it, reads from SRA, use the cloud, use faster QDump. Logan contigs are fast to download. They are in FASTA format, which is great. Logan search is finally the blast for SRA that I've been talking about for years at CGSI. It finally exists. Happy about it. And I leave you with this CGSI 2025 starter pack. And happy to take questions.